Welcome back to the course. So today we'll discuss about the continuation of the discussion on antigen presentation. So we completed our endogenous expression of the antigen. So today we'll discuss about the exogenous expression of the antigen and exogenous processing of the antigen through MHC2 pathway. So we discussed this endogenous pathway, and now what is exogenous pathway we are going to discuss. Just one thing which I would like to emphasize here to students is these two pathways are although critical in order to present the antigen, but there could be a way once the endogenous antigen may be presented through MHC2. Say this endogenous antigen may be presented through MHC2 pathway, while exogenous antigen may be presented through MHC1 pathway. That means there could be a possibility that the antigens which are processed through endogenous or exogenous pathway can load into a different molecule altogether away from the canonical binding affinity. This is called cross presentation. So this is cross presentation. So cross presentation stands for the presentation of antigen through different molecules. So endogenous can be expressed through MHC2 pathway, and exogenous can be processed through MHC1 pathway. This cross presentation of an antigen could lead to a variety of different immune response altogether. This property is related to the feature of an antigen. Some of the antigen can behave differently and all, which we will discuss in our future classes, but just for the knowledge. So we will discuss this cross presentation in our future classes. All right, we will move ahead to exogenous presentation. So to understand exogenous presentation, there could be a way to understand how these antigen presenting cells can present the antigen or a protein in a different way. So although this is related to the processing of antigen through different pathways, but this is a generalized phenomena. So for example, there could be three ways where the proteins can be tracked or trapped. So first is clathrin mediated endocytosis. So the clathrin mediated endocytosis is actually the cell invagination into the plasma membrane by these specific proteins called clathrin. So these are lining the vesicle. In addition to clathrin, these receptors such as lactin receptor or the endocytic receptor can trap the antigen, and these antigen can be processed by different proteases, and ultimately that can be presented through different pathways. If the antigens are in the soluble form, we called it cell drinking or pinocytosis. This pinocytosis may be called macropinocytosis when the amount of the soluble antigen is larger in volume, and once it is processed, then again the similar kind of proteases cleavage can happen, which can again lead to the processing of. So macropinocytosis can also lead to antigen presentation when the antigens are in the solution form. The third one is phagocytosis. Again, the phagocytosis is very specifically with a particulate antigen. So this could be a particulate antigen, while pinocytosis is very specifically the soluble fraction of the antigen. So the particulate one is actually engulfed by different phagocytic cells, and the receptor again can process this antigen. Sometime it could be a toll-like receptor expression, and then further the processing can happen through the antigen presenting cell. In addition to that, there is another pathway which we have discussed in the past called autophagy, which can lead to the self-degradation of the cellular content because there is a requirement by the cell, because the environment of the cells are not conducive. So these are the mechanism which cell does in order to maintain the homeostasis or homeostatic balance between the cell types. So this is just in rough that we need to understand there could be a clathrin mediated endocytosis, there could be a pinocytosis or phagocytosis. Now just one thing to emphasize here, that in research people always look into what kind of processing happening to a particular antigen. Let's say there are antigens which are called super antigen, there are antigens which are particulate antigen, how they are processed through the pathway, and whether these antigens are actually processed through one or multiple pathways can be studied using some inhibitors. So there are chemical inhibitors which are available in the market that can stop the phagocytosis or pinocytosis or say clathrin mediated endocytosis, and accordingly the scientists can study the way antigens are processed into the cellular system. So this is again a research based way to ascertain that how the antigens are processed into the cellular system. So let's try to go in the MHC2 pathway, which is called exogenous pathway. Now only one thing which we need to emphasize or which we need to remember is MHC2 pathway, the antigens are coming from outside and therefore we called it exogenous. Now so let's try to see one by one. So we will go in a different way here in the diagram. So let's try to see this panel first. So that's the first panel. Now here we can see that the MHC2 proteins are actually located in ER lumen. So one can see that there are two chains of alpha and beta of MHC class 2. These are present in the endoplasmic reticulum, and remember there are multiple antigens might be floating in the ER lumen. To avoid the entry of this peptide or this smaller protein fragment into this loop of alpha beta, there is molecule called Li, which is called invariant chain. Now this invariant chain can dock itself into this structure so that none of the other antigens can be loaded over the MHC. Otherwise we will see that there is a presentation of an antigen. To avoid the self presentation of an antigen, this Li which is an invariant chain can be blocking this loading area of MHC2 molecule, and this can be recycled through Golgi apparatus to the outside environment. And then of course this whole process is again recycled into early endosomes and then again recycled back to the peptide pathway. So there could be a phenomena where the MHC molecule itself is getting recycled through the cellular system, where the antigen loading area is actually occupied by Li chain, invariant chain. So this is important in order to understand that if there is no antigen present in the cellular system, there is still expression of MHC. So MHC is constantly present in some cell, particularly we are talking about the antigen presenting cell here, because this is MHC2. And these antigens are actually not allowed to be docked into alpha beta docking area because otherwise we will see an immune response, and therefore this Li chain does play a very important role in maintaining the MHC say recycling inside the cellular system. So this is one way. The second is when there is an antigen presentation. So once there is an antigen presentation, so this is the second pathway. So when there is an antigen which is there coming into the system and so this area is actually the compartment where antigens are processed. So we need to understand this pathway a little carefully, because this compartment, which is called intravesicular lumen or interluminal vesicle, ILV stands for intraluminal vesicle. Now intraluminal vesicles are actually the small vesicles which are present inside the cell and can help in loading of the peptide over the MHC2 molecules. Now just recapitulate the same thing, that MHC molecules which are there in the cells are actually occupying or the alpha and beta chain of the MHC2 is already occupied by Li chain. This Li chain is actually clipped off by specific proteases present in the endosome or this luminal vesicle, and because of that proteolytic cleavage, the only small fragment of the peptide will remain in this premature form of MHC2. We call it clip, which is actually a small molecule which are docked into the site, and these are the remnant of Li chain. So, remember, the clip is formed by the proteolytic cleavage of the Li chain in the endocytic vesicles or in the intraluminal vesicles, and these clips are actually will be present in the system till there is an antigen entry. So, once there is an antigen entry, the clip molecules are removed by a very specific kind of a protein called HLA-DM. Now, HLA-DM is another protein in the vesicle which can remove the clip and load the antigen properly. So, now this antigen which are supposed to be there will be loaded through this removal of clip by HLA-DM. So, HLA-DM is again a protein which can remove the clip and allow the entry of antigen to be docked into the MHC class 2. There is another molecule very close to HLA-DM, is called HLA-DO. Well, the function of HLA-DO is to maintain the homeostatic or the functionality of HLA-DM. So, DO can help the activity of DM, and DM can remove the clip molecules in order to dock the antigen. So, this is again a very synchronized and complex phenomena that is happening in the vesicle, where the antigens are actually loaded post removal of the self generated Li chain specific clip molecules. So, clip is removed, and now these peptides are loaded on MHC1, and then this can be trafficked to the cell surface. Most of the time this can be identified by this specific CD4 specific immune cells. So, these are T cell, and these T cells are called T helper cell. So, this loaded peptide on the MHC2 can be identified by a very specific T helper cell population, which is positive for CD4 expression. So, this is CD4 specific T cell population, which later on will produce a B cell immune response, or say antibody production can happen through this pathway. So, this is actually the way these antigens are processed through the MHC2 molecules. So, we will go in just in nutshell in the next slide, but this is the second pathway where the antigens can be loaded with the help of this HLA-DM and DO molecules. Now, once these antigens are loaded on the MHC2, there could be a phenomena where this could make a small exit by these exosomes, and these exosomes are a small vesicle again carrying this MHC2 loaded peptides. This could be circulated into the body cavity or in the body system, because this is just an extracellular fluid or extracellular space where the cells can float independently and do some activity related to cellular biology. Now, these exosomes are again important because these are carrying this MHC2 specific peptides can be transported to different areas in the cell, and then probably they can modulate multiple different pathways. So, this is the third one where we need to focus on. So, the first one is recycling of the MHC2 molecules through the Li chain, and second is actually the processing of the foreign peptides over the MHC2 with the help of the HLA, DM and DO molecules, and of course, the removal of clip and the Li cleavage by proteases. And finally, there could be a possibility that if there is no expression through or no docking through T helper cell CD4 plus T cell, then the MHC1 loaded vesicle, the small vesicle, can secrete out in the form of exosomes, which are again a very critical. In recent years, the biology related to exosomes, the specific way exosomes are targeted for diagnostics and disease prediction is coming up or flared in a very large scale. So, let us move on and see exactly the MHC2 MSC2 pathway. Again, in the same way that antigens which are trapped through the extracellular space, most of the time these are bacteria, and this bacterial population is now once entrapped can be processed by endosomal acidic pH, and this endosomal acidic pH can go to the area where the peptide can be docked into the MSC2 molecules, and this MSC2 is again trafficked through the extracellular space in order to process it through CD4 specific T cell. Now, let's try to understand that the CD4 specific T helper cell can have the presentation of this antigen and can subsequently raise a B cell mediated immune response, or we could say antibody mediated immune response in future if it is processing and activating the B cell population. Now, the antigen presenting cell here will have the invariant chain loaded MSC2, which we have already studied, that this can be cleaved into small clip fragment by proteases, and then this later clip will be later pinched off by HLA DM molecules present in the endosomes. So, essentially this is a very different pathway as compared to the MHC1 expression, where we have seen that the MHC1 loading is happened because of chaperones present on ER, and these chaperones can first hold the MHC1 molecules, and later on it can help to dock the peptide with the help of tapacine. But here the things are different. The MHC2 molecules are already loaded with a peptide called clip, and once clips are removed with the help of HLA-DM, then the peptides, the external peptide or the non-self peptide can be loaded over these MSC2 molecules. So, this is a gross way we need to understand about the exogenous pathway, and remember that this is CD4 specific T cell immune response, because here we are activating the CD4, and there in MHC1 we are activating the CD8 specific T cell, which is also called a cytotoxic T cell, whereas this is called T helper cell. So, let us move on and see a little more about the invariant chain, again emphasizing the same points on its cleavage with a little more elaborative cartoon like figure. So, this is the actual location of the clip. So, clip can be loaded in a way so that the whole area can be occupied, and because of the acidic pH, this fraction can be cleaved out, and then we end up in getting only the small clip fragment, and this clip is later on removed by this HLA-DM molecule in order to give entry to an external antigen. So, very synchronized way the loading is happening in MSC2 molecules. So, this we need to remember that the class 2 is actually clip based phenomena, class 1 is actually tapacine or tap specific phenomena, because the transporter of antigen, the tap protein present on the ER of antigen to the luminal area. So, this is how these invariant chains are processed. So, this is about the final pictorial representation of the activation of CD4 T cell by MSC2, and what is happening here is a dendritic cell expressing the antigen through MSC2 can bind to the TCR, which is a T cell receptor present on T helper cell, and this is again a stimulation pathway which we will study in the later classes, but just remember that these are other accessories protein which are required to facilitate the expression or activation of T helper cell in order to do multiple functions. So, the activated T cell or T helper cell can produce lot of different cytokines. Some of the cytokines could be interferon gamma or interleukins, which can activate further pathways and downstream signaling to give an effect to the immune system. So, one more thing which we need to understand is how this redistribution of MHC class 2 can happen into the system when the antigens are present to the mature or immature dendritic cells. So, what we need to understand that the recycling of or redistribution of MHC2 molecules are done with the same loading structure. So say we can say that the MHC2 molecules are loaded with Li chain, and this Li chain is again cleaved by the proteases present, and then this can go on the surface and present the cell peptide. Now these are specific activities which can happen when the mature dendritic cells are not mature. So these are immature dendritic cells, where the self antigen can be processed by basically removing this clip molecules and loading the peptide molecule. Second, when there is a pathogen entry inside the system, then the activation can happen in a way when the clip molecules are removed in order to dock the foreign molecule. So here the antigens could be self in nature, here the antigen could be non-self in nature, and still we are seeing the expression of the antigens by MHC2 molecules, while once there is a possibility that T helper cell population can be present in an area. So these naive CD4 specific T cell can identify this foreign molecule and can start the signaling cascade to start the immune response, which are antibody mediated immune response. So this could be a mature dendritic cell. So one can see that the expression of the cell peptide, expression of the non-self peptide, and expression of an non-self peptide loaded over the MHC1 once bind to the naive CD4 T cell can give a mature dendritic cell ability to activate the antibody mediated immune response. So this is all about the redistribution of MHC class 2 molecules, which are again very important in order to process the antigen by dendritic cell population. And we need to see that the expression of these MHC peptides on the surface can give an indication that whether the dendritic cell are mature or immature, or whether these are maturing dendritic cell. Say they have not attained the ability to bind to the CD4 specific T cell population. So we will try to see the actual pathway and all those antigen expression pathway, how this can modulate the immune system in total in our next class. Thank you.